Assalamualaikum and a very good morning. I bid to all the judges, lecturers and fellow colleagues. I am Syaza Amni and my partner Muhammad Rafizul Helmi will be presenting our research regarding antimicrobial effect of Heterotrigona itama propolis from two different regions in Malaysia against Faecalis. It is supervised by Associate Professor Ikmal, Professor Dr. Fouad and Dr. Hasnah.
First of all, as we all know, endodontic infections are a common complication of endo-treated teeth. E. faecalis is the most frequent microorganism found in failed root canal treatment. A study by Pimenta et al. In 2015 suggests that E. faecalis is resistant towards calcium hydroxide, which is a common intracranial medicament used in root canal treatment. Calcium hydroxide is used due to its antimicrobial properties, osteogenic potential, and temporary physical seal it provides towards the root canal. Although E. faecalis can withstand and survive in this environment created by the calcium hydroxide. Moving on to propolis, which is also known as bee glue. It is formed with the reaction of components between trees and plants with bee secretions such as wax and saliva. It contains more than 300 components such as phenolic compounds, flavonoids, and flavanones. Their composition depends on the regional climate, the type of trees accessible to the bees, and some depends on bee species. The dominant pollinators for Peninsular Malaysia includes Heterotrigona itama, HI species, and Geniotrigona thoracica, GT species. In this study, we decided to use HI propolis because previous studies have shown that HI propolis contains higher amount of flavonoids when compared to GT propolis. To further explain, different regions with different type of trees plays a major role in determining the propolis content. This table on my left shows the farms in Raup and Lenggong with different types of trees.
The problem statement in this research is Faecalis has a greater resistance to the calcium hydroxide. In many cases, full debridement of infected canals cannot completely disinfect some areas due to the anatomical complexities. As we all know, this intracanal medicament is synthetic product and cytotoxics. Therefore, alternative agents need to be found, especially natural products such as propolis. Studies have shown the potential of propolis against resistant microorganisms. Until now, there is no research comparing the antimicrobial properties of Malaysian HI propolis in different regions. Our research objectives are to determine and compare the antimicrobial activity of Malaysian propolis produced by HI from Raup and Lenggong against Faecalis.
For the methodology, we purchased the propolis from Humayrat Honey Sendirian Berhad in Lenggong and Asta Stingless Bee Honey Farm in Raup. Then, we extracted the propolis samples with 5 steps, which are dissolution with 70% ethanol, sonication, filtration, evaporation, and lyophilization for 3 days. For the preparation of bacterial suspension, E. faecalis coded ATCC29212 was purchased from Sigma Aldrich. Then, it is rehydrated in brain heart infusion broth BHIB and is left incubated for 18 to 24 hours. Glycerol stock were also prepared from the BHIB samples and is stored in negative 80 degrees Celsius freezer for further use. Next, we proceed with antibacterial assessment, which consists of 4 tests. The tests are antibacterial sensitivity test, AST, minimum inhibitory concentration test, MIC, minimum bactericidal concentration test, MBC, and scanning electron microscopy, SEM Analysis. For the data analysis, Kruskal-Wallis test was done on AST result and repeated measures ANOVA was done to compare the results from MIC.
As for the results that we have obtained, the AST test shows 100 mg per ml of MLP have a maximum zone of inhibition with diameter median IQR of 16 mm. The value of MIC estimated based on naked eye observation. The MIC value for MRP was 1.56 mg per ml, while for MLP was 6.25 mg per ml. Next, the results for the MBC, which MRP extract completely inhibited the growth of Faecalis at the concentration of 25 mg per ml, whereas for the MLP, it was observed at the concentration of 100 mg per ml. This graph shows mean absorbance of MIC of MRP and MLP extract against Faecalis that was analyzed using repeated measures ANOVA. Based on the statistical analysis, the overlap data at similar concentration showed no significant difference, whereas with no overlapping data, significant difference was seen between the concentration. Last but not least, SEM analyses were done to evaluate the cell morphology of Faecalis after treatment with MRP and MLP and compared to the positive and negative controls. It shows 5% DMSO exhibited normal morphology Faecalis. However, the cells treated with MRP and MLP extract showed changes in the cell morphology. The effects of the MRP on the cell morphology was more evident compared to MLP. Positive control calcium hydroxides was also found disrupting the cell morphology. The view of the most of the bacteria were obscured by the presence of the sedimentation caused by the calcium hydroxide.
For the discussion, AST showed MLP has a greater inhibition zone compared to MRP, while MIC, MBC, and SEM analysis showed that MRP has a stronger effect on E. faecalis compared to MLP. This may be due to the activity of the active components and compounds present in the extracts. Furthermore, AST only compared the effects of samples at certain concentration, while MIC and MBC were done to obtain the lowest concentration to inhibit growth and completely kill the bacteria respectively. SEM analysis showed that MRP has a stronger effect on the bacterial cell morphology compared to MLP. These differences could be explained due to the various and different types of trees surrounding the beehives. Studies by Tira Siripricha et al. In 2012, Kustewan et al. In 2017, and Halid et al. 2019 stated that propolis from two different regions of two different bee species during two different harvest times contain a component named cardol. Cardol can be found in Anarcadiaceae tree species, which is Cateria trees present in their farm. Previous studies by Shahata et al. In 2020 and Ferreira et al. In 2017 states that different types of plants produces variations in their bioactivity and physiochemical contents. A study by Mahani et al. In 2013 implied that different propolis from the same species from different regions of Java and Sulawesi shows different anti-diabetic properties. Although a study by Ibrahim et al. In 2016 states that both factors plays a role in determining propolis content. For Malaysian studies, which are two papers by Ibrahim et al. In 2016, Azamin et al. In 2018, and Manafi et al. In 2019, all stated that it is bee species dependent and that HI propolis is better than GT propolis in terms of antioxidant activity. The source for these propolis were taken from Besut, East Coast in Terengganu. Meanwhile, a study by Asim et al. In 2020 states that GT propolis is better than HI propolis in terms of antioxidant activity. The source for these propolis were taken from Bertam West Coast in Pulau Pinang. Based on these findings, we can say that due to the different regions the propolis were collected from, the results of the findings were different. This further supports our hypothesis that the types of trees does influence the chemical composition of propolis. For our study, we can conclude that MRP shows a higher antimicrobial effect compared to MLP. This is shown by MRP requiring a lower concentration to inhibit the growth of E. faecalis compared to MLP. SEM analysis also shows that MRP has a more profound effect on the bacterial cell morphology compared to MLP. Therefore, we can conclude that HI propolis from Raup and Lenggong are different due to the different types of trees and plants accessible to the bees surrounding the beehives. For our recommendations, firstly, for SEM analysis of calcium hydroxide, we recommend using other types of methods of comparing effects on E. faecalis, as calcium hydroxide is prone to sedimentation, resulting in an unclear photograph. Secondly, further studies on which contributing trees provide specific antimicrobial properties should be done to confirm which component provides which specific properties. And lastly, further studies on how geography predominates over bee species in determining the propolis content should be done. Our effort is to maintain natural and sustainable resources, as well as invigorating the local existing Malaysian apiculture. And with that, we end our presentation today. Thank you to the audience for spending your valuable time listening to our research presentation.